The Asheville Science Tavern Second Sunday for Science event. My name is Elisa Hovey. I'm a biologist and I'm a professor at Warren Wilson College here in Asheville. And it's my pleasure to welcome everyone to to our speaker series for tonight. Just to kind of give you a little bit of background, the Asheville Science Tavern, we're a social informal group of folks who often meet at White Labs in downtown Asheville. But for now, we're meeting on Zoom, and our goal is just to really connect scientists in our area and beyond with members of the community in a friendly and informal setting where we can ask questions and talk with one another. So that's what we're here to do tonight. And it's really a pleasure to introduce our speaker tonight, Dr. Langdon Martin. Langdon is a professor of chemistry at Warren Wilson College. He is a colleague. We came into Warren Wilson College in the same year, and he's someone who I've come to consider as a friend and a really inspiring person when I think about connecting people to science. Langdon's background is in organic chemistry, and since coming to Warren Wilson, he has really embraced the spirit of teaching and trying to integrate students into his work and developing new ways to take products that are produced and develop them in ways that can be integrated into environmental sustainability initiatives. And without any further ado, I'd like to invite Langdon to share his screen and share his research with us. So I'm going to mute myself now, and Langdon, thank you for joining us tonight. I really appreciate you.
Thank you. Thank you, Elisa. Can you all see my slides now? Thumbs up. All right, great. So thank you all for coming. And yes, it's my pleasure here to talk about some of the research. As Elisa said, I've been incorporating as many students as I can into this. So a lot of this is really their work, and trying to talk about stuff that's going on and relevant to Asheville. So I guess to start off with, I want to, sorry, there we go. I want to thank the folks who've really done a lot of this research. So first, my colleague, Rusty Bryant, who's been collaborating on this broader project since its inception about 6 years ago. And then really there are 4 or 5 students who've been on this project. Rebecca Truitt got this started about 5 years ago. And then the research I'm going to be presenting tonight are from Virginia Shukovsky, Reese Burns, and Chris Fadler-Cree. And then there are some students who are taking up this project, and maybe we'll give another talk in another year or two with some follow-up. Also, Michael Flythe, who works for the USDA and the University of Kentucky. And then a lot of this work is in collaboration with an Asheville area brewery here. So this is the Asheville Science Tavern. And so it seems appropriate to talk a little bit about beer. I'm going to start this talk trying to kind of teach you all a little bit more about the beer brewing process and some of the chemistry that maybe we don't necessarily think about as much with the beer brewing process, but that may actually have a hand to play as we fight climate change and try to mitigate global warming here. So there are a lot of breweries in Asheville. I think over 50 by the last count that I saw. I don't know exact number here, but we were Beer City, USA for a number of years. So there's a lot of craft brewing going around Asheville. So these are smaller places. They make smaller batches, try different flavors, that sort of thing. And that's going to be important later on here. And what this talk is really going to be doing, I say upcycling on this on the title here, is taking a byproduct or a co-product of the beer production process and seeing if we can turn it into something that's actually valuable and maybe can help us mitigate climate change. So Here, this slide here just shows you a little overview of the beer brewing process. And at top, what I've got here, there are really only 4 main ingredients to beer brewing. There's water, there's malted grain, there's hops, and there's yeast. So in the first couple of steps, you take your malted grain, the grain that started to sprout, started to turn some of those starches into sugar. You mill it, you mix it with a whole bunch of water, and you heat it up. You get some of those sugars to start dissolving in the water, and that becomes the sweet mixture called wort. When that is done, you've got your wort, your sugar water solution that's going to eventually become the beer, and you've got spent grain waste. That's a co-product, but that's very well known. Pretty much all breweries have a use for this. This is often sold or given to farmers. So the spent grain here, it's very protein rich. It's still got a lot of cellulose. So a lot of livestock, I've shown goats here, but there are others. A lot of livestock can eat this. So again, it's kind of a win-win for farmers. They get food. Breweries have a waste product that they have a good way to get rid of, and that gets repurposed to good use. So after the wort's been made, then we add the hops. Heat that up, start extracting all the good stuff from the hops. I'm going to talk more about that in a few minutes here. Then you cool that back down to a temperature where yeast can live. You add the yeast and you ferment that for a while. At the end of that fermentation process, the co-product, the byproduct that's formed is something called spent yeast. And that's what I'm going to be spending the rest of this talk talking about. So the spent yeast is usually something around 5% of the total volume. So, you know, they're made, if you've ever been in a brewery, you've seen these giant vats where they have dozens or hundreds of barrels of beer fermenting at any given time. Roughly 5% of that volume of beer production winds up as this product called spent yeast. So if you think about it, you get 3 6-packs of beer, that's 18 cans of beer. There's also a can, so to speak, that the brewery has of spent yeast that it's got to deal with. And so it, you know, just gets drained out of the bottom of the vat here, and it's this kind of this funny looking paste. It smells kind of nice. It smells like beer. It's very thick. It's kind of like maybe like a thin peanut butter is how I would describe it. So, and it's by volume, there's a lot of water there, but there's yeast. Yeast is interesting in that it's a complete protein. So the large multinational breweries, your Millers, your Budweisers, and what have you, actually have a good use for this co-product. They can put it into tanker trucks, haul it away, evaporate all of the liquids off it. They can sometimes harvest the ethanol, and then they wind up with this dried yeast powder, and then they can sell that as nutritional yeast. They can turn it into pet food, into Marmite. They have uses for it. These smaller breweries, the craft breweries like the ones that we have in Asheville, they don't quite make enough of this stuff that it makes sense to haul it away in tanker trucks. So they basically have 2 options. They can compost it, or they can send it down the sewer. It's a bit of a problem going down the sewer. They have to pay even the sewage company, because there are very specific microbes that are treating the sewage. And if you're throwing this yeast paste, which contains live yeast, that can throw things off. And so if we can find a better use for this, the breweries will be happy. So that's what I'm going to be talking about now. So the title of my talk, I said, let's hop to it. reducing agricultural methane. And the reason I said that is because the spent yeast that you saw there contains a lot of the flavor and the fragrance and the taste molecules that went into the beer from the hops. So earlier on the beer production side, you saw it, you added the hops, and then soon after that, the yeast came in. So there are a bunch of really interesting molecules that come from hops into beer. As Elisa said, I'm an organic chemist, so you're not going to get through a talk with me without seeing a few line angle chemical structures. So there are some terpene-derived molecules that you can see on the left here and these are responsible for a lot of the fragrances that give beer its smells and its taste. The other molecules that come from the hops are something called the hop acids, and you can see the alpha acids and the beta acids, and they provide a lot of the bitter taste in beer. And the alpha acids will actually even isomerize during the beer brewing process and form what's called the isoalpha acids, which are even more bitter. So we humans appreciate the hops for their flavor and fragrance molecules, but the reason that these were added to beer, well, the reason we add these to beer is for their flavor and fragrance, but there's another interesting thing about hops, and that is that they have some antimicrobial effects. I guess what I want to talk about first, before I really get into the antimicrobial effects, is say, again, so we've got all these structures, all of these interesting organic molecules that wind up in beer. They're hanging out in the beer during the fermentation process. Some of them wind up in the beer, some of them wind up in the spent yeast. So again, here's your chemistry lesson for the day, is if you look at all these molecules, they've got a lot of carbon and hydrogen. So the lines and the angles all represent carbon and hydrogen. They're very hydrophobic. Whereas beer is mostly water with a little bit of ethanol in it. And so a lot of these molecules do wind up in the beer. That's most of the volume here. But yeast, it's a microorganism. It's got cell walls, lots of lipids and so forth that are really good at dissolving these aroma and flavor molecules, these bitter tasting molecules. So a lot of these interesting molecules from the hops don't wind up in the beer that goes into the bottle. They wind up in the spent yeast, and that's really important for what we're trying to do here. So now let's talk about a little bit about microbiology. So the hops here have these, I'm going to be speaking now mostly about the alpha and the beta acids, so the structures that I've shown on the right. So these are what are responsible for the bitter taste of beer, but they also have some really interesting antimicrobial effects. So they will kill microorganisms. And that's probably why the hop originally evolved these secondary metabolites. And so what my collaborators, Rusty Bryant and Michael Flythe, did was this bacterial lawn that I've shown you here. And this is what really kind of got us excited about doing the research that my students have gotten in. So on the right here, what you see here is a petri dish with some bacteria growing, and the bacteria in this turn kind of this reddish brown color. In the center, they put a spot of baker's yeast. That didn't affect how the bacteria grew. On the left here, they used beta acids, so some of these lupulone molecules on the right, that really inhibited the bacteria from growing in the center here. And then in the center, Petri dish here, you've got the craft brewer's yeast. So you've got yeast, which doesn't on its own inhibit bacterial growth, but because it's absorbed these beta acids, these alpha acids, these isoalpha acids, it does prevent some of these bacteria, some of these microbes from growing. And in fact, I mentioned, I think I meant to mention this earlier, but I didn't. Oh no, here's where I wanted to mention it. So another thing to note about craft brews is they like styles of beer that often have a lot of hops. So if I zoom in, particularly on these here, the top 5 styles of beer that are brewed by craft breweries include India Pale Ale, that's the #1 style. And then number 4 here is the American Pale Ale and the double IPA or the triple IPA. And that can have, as you know, the IPA, the India Pale Ale has a lot of hops in it. The reason that India Pale Ale got its name is because the British, when they were shipping beer to India, figured out that if they put a lot of hops into this beer, it made it really bitter, but it would survive the long ocean voyage to India. We now think we're pretty sure that's because these hops imparted these antimicrobial molecules to that beer. And so now there are double IPAs, which have even more hops in them. American Pale Ale has quite a bit of hops in them. Your Budweisers and your Millers of the brewing world, they have a little bit of hops in them, but not really enough to show these antimicrobial effects. So it's great for us that these craft breweries use a lot of hops in their beer. That makes their spent yeast have a little bit more antimicrobial potency. All right, so now I get to really talk about the research that 3 of my students worked on in the last 4 years or so here. So Chris worked along with Rusty, and so what they did was took a few different spent yeast from a few different beer styles from our brewery Collaborator. And they extracted the hop acids from them. So what you're seeing here with the green peaks or the green bands here is an HPLC trace. And so they, I won't go into all the details about HPLC, but it's a way of taking a complex mixture and you run it through a column. Things move through the column at different rates, and it allows you to separate and analyze them. And the other thing that's nice about HPLC is you can figure out, with the, depending on how big the peaks are, depending on what the area is under the curve, you can actually use this quantitatively, figure out how much you have. So you see a couple of the alpha acids come off first here and the beta acids come off a little later. And then in the bottom left here is our standard curve. So Chris was able to say, okay, yes, based on how much alpha acid, how much beta acid I have in the spent yeast, I can quantify, or I can quantify how much I have in the spent yeast. So he looked at 4 different beer styles and made this this chart here. So we're just going to call them A, B, C, D. You can see there's a lot more alpha acids, those blue bars, and then the orange bars are the beta acids in the different beer styles. And then the gray bars are essentially just the sum of the blue and the orange bars. And what he also found that was interesting is that correlates pretty well, not perfectly, but pretty well to the amount of hops that the brewery originally put in to the beer. So you'll notice here the graph on the right here, it doesn't have numbers on the y-axis. The brewery didn't want to give away or doesn't want me to give away their hopping secrets, but you'll have to take me on faith here that this is true data, I guess. But these different hopping rates that the brewery puts in is what's on the y-axis, and then the x-axis is how much Chris was able to extract from them. And so you can see there's a pretty good correlation here. So C was probably an IPA or a double IPA, and then A was something that maybe just a pale ale, maybe something even less like a Pilsner. All right, so I've been talking a lot now about beer and hops. And what I promised with the title is that we were going to talk about mitigating global warming as well. So let's tie this together here. So many of you may be familiar with this information, but it bears repeating. So the US releases quite a bit of greenhouse gases. About 10% of it, shown on the pie chart on the left here, is from agriculture. Some of that is carbon dioxide. That's kind of the first gas that we think of. Methane is another one. And methane is about 20 times as potent a global warming gas as is carbon dioxide. And methane is one of the big players in terms of agricultural global warming. So of that 10% of agricultural greenhouse gas emissions, a lot of that comes from cattle. So roughly a third of that comes from cattle and cattle waste. Not a whole lot of that is carbon dioxide. Most of that is actually methane. So the Americans drink a lot of milk, we eat a lot of cheese, we eat a lot of cheeseburgers. And so cattle are a huge contributor then to agricultural global warming. So cows emit a lot of methane. The reason for that is they have a really fancy, well, maybe not fancy, but sophisticated digestive system. They're called ruminants because we sometimes talk about them having 4 stomachs. They have a much different digestive process than we do, which is important because they can eat grass. So when they eat the grass, the first place that it goes is the rumen. And inside the rumen, they have a ton of symbiotic microorganisms, including bacteria, including archaea. And those are able to break down the cellulose in the plant material and turn it into things that the cow can then absorb and use. Part of that process, part of that fermentation process, there is methane is produced as a byproduct. And when methane is produced, the cow just has to burp it out and it's released into the atmosphere. So methane production, it's probably not something we can completely stop, but if we could lower it, that would be great. That would do a lot for reducing global warming. It also would be good because of this thing called feed efficiency. So carbon is a component of fat as well, and sugar. So all of this carbon that's leaving the cow as methane is not becoming milk or beef. And so if we can lower the amount of methane that's produced, potentially then we're going to increase feed efficiency. So of the feed that the cow eats, more of it will become cow or milk, which would be great for the farmer, to say nothing of the environment. So to do this research at Warren Wilson, we had to sort of mimic the rumen at least a little bit. What's interesting about the rumen, what's challenging is that it's an anaerobic environment. And all of the methanogens, the methane-producing microorganisms, are archaea. And these archaea cannot survive, they cannot grow if there's oxygen around. So we needed a fairly simple setup that would allow us to remove oxygen. So what we've got in our research lab here is what's called a glove bag. It's this giant, thick plastic bag where there are essentially holes with gloves where you can put your arms and manipulate things in there. and you fill it with argon and or carbon dioxide and exclude the oxygen. And that is close enough to the rumen that these archaea can now continue to grow and produce their methane. And then we actually do these growths, you can see, in little jars, as you would with probably any bacterial growth, but they've got a rubber septum on the top. So again, we fill them in the glove bag here, tighten them. Now we have an anaerobic environment. And every now and then we can just stick a needle, a syringe into the septum and pull out the atmosphere and see, okay, how much methane is in there. And so that, Rebecca got this started, and then that's what Virginia and Reese did for their research. So again, to look at the methane, we used another type of chromatography. In this case, we used gas chromatography. So we took the methane out of the well, we took the entire atmosphere out of our growths, and then we put it into the gas chromatograph. So again, separated it from the different types of gases, and then are able to look at it using a detector. So what the graph on the left shows is just one of our proof of principle types. Okay, we can get this to work. We just took some natural gas from the gas valve. And that's where the methane and the ethane come from. We also just kind of breathed into the bag, so you can see the carbon dioxide peak there. And that's really those are the main things that we're going to see from these growths. Same idea here on the right. You can see our standard curve. There's a nice linear relationship between how much methane we inject into the gas chromatograph and how big the area is under the peak. So one of the first experiments that Virginia did when they started this project was to just take some rumen fluid and take some rumen fluid here and some buffer, and quote unquote grow them for overnight. And when they did that, we got this graph here. So you can see, okay, when you mix rumen fluid, so you mix rumen fluid, buffer to mimic the rumen, and then we used just a little bit of fructose as the sugar substrate. When we did that, when we added rumen fluid, we got a whole lot of methane. When we didn't add any rumen fluid, we didn't get any methane. So we had a nice proof of principle here. So then what Virginia did was to get some rumen fluid first from a cow and treat it with buffer and with fructose, and in the presence either of baker's yeast or in the presence of craft yeast. So we used baker's yeast as our control. So essentially they're both getting the same amount of protein. But so the only thing different here should be the presence or absence of the molecules from the hops. And what Virginia saw was that there was roughly a 28% decrease in the amount of methane produced when there was craft yeast present. Then what they did was to go and visit our collaborator, Michael Flythe, who had fistulated goats. So I should mention the microbes from the cow. Virginia went to the abattoir, the slaughterhouse, and they just gave them the rumen from a freshly slaughtered cow, and they went in and collected some of the rumen fluid and did that for our experiments. Then Virginia went to Kentucky visited Michael Flythe's labs and did this with some fistulated goats. And so in that case, we were able to take some rumen fluid out without harming the animal. And with the goat microbes, also Michael Flythe has a much nicer setup than just a glove bag. They were able to see a 61% decrease in the amount of methane produced when there was craft yeast as compared to baker's yeast. And so we published that in that Advances in Microbiology paper there. So that was Virginia's research, and where they finished up, Rhys Burns picked up. And so again, you've seen this slide before. The cow has an interesting digestive system. So Rhys wanted to continue this, this methane work. She also wanted to look at ammonia. So ammonia is a byproduct formed in the rumen. It's not involved in global warming, but it still is involved in feed inefficiency. Nitrogen that becomes ammonia doesn't stay as protein in the animal, which is what the farmer would like. And it also can, if you form too much ammonia too fast, it can actually kill the animal or make it quite sick. So if the craft yeast will reduce the methane, will reduce the ammonia in the animal, that would be great. The other thing that Rhys wanted to compare with is an antibiotic that is used by farmers generally these days, something called Monensin or rumensin. So the information on this slide is just taken from the website of Elanco, which is who makes a lot of the commercial rumensin. So this is an antibiotic that is normally given to cattle, particularly cattle in feedlots, because it can help improve feed efficiency. And so we wanted to see, okay, we're expecting that this will probably, the rumensin will reduce methane. We wanted to see how did it, and how does that compare to the spent craft yeast, which has all the hop acids. And so again, yeah, so what Rhys looked at was microbes and looking at craft brewer's yeast, baker's yeast, Monensin. Here's a picture of Rhys. I get a kick out of this picture whenever I go by it in the hall now, because this was taken 4 years ago before coronavirus, but here she is wearing a mask because it was good too for this work that she was doing. So she looked at these three different experiments, and she also switched. So Virginia used fructose as a substrate. Rhys switched to ground hay as a substrate because that's more similar to what the animals actually eat. And so here, I won't go through these results too much detail, but just to give you an idea, she found some really interesting things. So you see, if you look on the left here, the blue is her methane results. So you've got how much methane is produced by baker's yeast, how much methane is produced by some growths with craft yeast, and then how much methane is produced in growths that include baker's yeast and the Monensin standard antibiotic. And what she found was that, as we would expect, adding Monensin reduced the amount of methane produced at a statistically significant level when Monensin was added. Also, kind of following up, corroborating Virginia's results, Rhys found that when there was craft yeast versus baker's yeast, there was less methane produced. There was not a statistically significant difference between craft yeast and Monensin. So this is exciting because it seems like maybe this is still a maybe, but down the line, if we can keep using this spent craft yeast, maybe we wouldn't have to give cattle this single antibiotic. We would be able to give them this other product and still get these same methane reduction results, potentially increased feed efficiency results. So that was the methane. The other really interesting thing that Reese found when she looked at the ammonia is that the only treatment here that really reduced the ammonia was the craft yeast. Baker's yeast, it was still high. Monensin, you can see it reduced it a little bit, but not to a statistically significant level. The only real difference here was, yeah, the craft yeast reduced the amount of ammonia produced by the rumen fluid with the ground hay substrate. And so again, this is exciting because if this works as a feed substrate, it could help decrease the amount of ammonia byproduct that's produced. That's good for cattle health, and that's good for feed efficiency. So one thing then that Rusty did, so Rusty worked with all of these students, and one thing he did to kind of follow up with Reese's and Chris's study. So again, I showed you this this chart earlier with the different amounts of hop acids that are found in the different types of yeast. And Rusty did an experiment then with these different spent yeasts and growing them. And what he found, was that the amount of methane produced is fairly similar to the, or how do I say, correlates well with the amount of hop acids. So you can see B and C had the most hop acids and they had the most inhibition of methane production, whereas A and D had very little inhibition of, or less inhibition of methane production, which is what we would expect because they had less hop acid content to begin with. So in summary, spent craft yeast, right now it's a brewery waste product. It just goes down the drain. They have to pay to put it down the drain. But what some of our research has shown is that it has potential to be, shall we say, upcycled into an animal feed. Yeast is high in protein, but it's also got spent yeast from craft breweries have a lot of these hop acid natural products in them. And based on my students' research, it looks like that will reduce generation of methane and generation of ammonia in vitro due to the presence of these hop acids. So some of the ongoing work that my current students are doing is one, so I've been talking in this talk about the alpha and the beta acids, the bittering agents. The spent yeast also smells like these flavor fragrance molecules. And so one thing that we're going to look at too is to see if we can recover any of these from the spent yeast as well, because they smell nice. They actually might have some commercial value depending on how much we can get out here. So we can distill these off, and you can see then we can run them on the GC and identify how much we have. The other thing we're interested in learning more about is, okay, spent craft yeast, these hop acids seem to reduce, or they do reduce methane production. Why? How? What? The rumen is full of many different microorganisms. What is it that these are affecting? Are they affecting the methane-genic archaea directly? Are they affecting some of the other bacteria in there that, say, produce the hydrogen that is used by the archaea to make methane? Is it something else entirely? So we're starting to look at that. Again, like I said, the rumen is very complex, so that's going to take a lot of work, but it's something we're very curious to find out. So with that, again, I would like to thank my collaborator, Rusty Bryant, who's been in this with me since the beginning, doing some of these experiments and helping me mentor all the students. Joe Young and Matt Burleson have been lab managers here. They've helped maintain these HPLCs and GCs. I talked to you about former students. Dania, Ashley, and James are current students who've done stuff on these projects. I'd like to thank our collaborators, particularly Michael Flythe of the University of Kentucky, also my colleagues in the natural sciences, including Elisa, for helpful discussions, my family, Anna, Eliza, and Lucille, and funding from Warren Wilson College, the Warren Wilson College Sutherland Grant, and the USDA. With that, I'd be happy to take a few questions.
Great, thank you. We do have a question from the comment section from Sean. And Sean asked, see, I gotta scroll to it. He asked, so if I have a septic system, am I putting my system at risk when I rinse my spent brewer's yeast down the drain in my sink?
Oh, I see. My guess is no. I mean, I guess that's a good question. I don't know that I know enough about septic systems and microbrew and yeast per se. I mean, I guess my gut would say I probably wouldn't worry about it too much. I know we have a septic system, and we put some microorganisms, you know, they come in like a little pack, and you put them in down the sink, you know, once every month or two. So I probably wouldn't worry about it too much, but that's a good question. I don't, that's a gut answer, but I really don't know.
All right, we have another question.
Yes, can you hear me?
Yes.
Oh, good. So my question is, do they feed cattle baker's yeast from the industry? Is that typical or not?
Not to my knowledge. I think so. Make sure I understand your question. So, you're talking about baker's yeast.
Baker's yeast, exactly. Because your research compared baker's yeast with the craft brewing yeast. So
Right.
do they typically feed baker's yeast to cattle?
Oh, I see what you're getting at. Okay, no. So what we would be, if we were to if we were to feed spent craft yeast to cattle, they do give various protein supplements to cattle. I don't think that baker's yeast is necessarily, or yeast in general, is necessarily used as a common one, but it's a complete protein. I think that's what makes it a good supplement. There shouldn't be any harm. You know, some of our collaborators have actually started some feeding trials, and there's no harm in feeding yeast to them. I don't think it's a standard practice, though.
I just wondered why you used the baker's yeast, compared that to the craft brewer's yeast. I just wondered if.
Oh, okay. So we essentially wanted to compare yeast that didn't have the hops in it, that didn't have the hops natural products in it. So the baker's yeast and the craft yeast are similar enough in terms of like protein content that it made a good comparison. So that's why we chose baker's yeast as our control there. Yeah.
Okay, thanks. And then I have another quick question.
Yeah.
How much craft yeast does a typical brewery, let's say like High Wire Brewery, make in their processing?
I don't know I couldn't give you a volume off the top of my head here. I'm sure our brewing collaborators could. Yeah, I don't know the answer to that.
Well, my question was, would it be kind of efficient for some small farm place, like some of these organic farmers that we have around here, our organic dairy farmers, would it be good for them to like link up with High Wire or some of these breweries around here to get some of this craft yeast, or is it not enough that would make it worth their while?
Right. So that's exactly the hope, sort of longer term here, is that if this spent yeast does turn out to be both safe and useful for cattle or any other animal, yeah, I would say most breweries, High Wire, anywhere, already have some sort of relationship with farms for their spent grain.
Mhm.
So it's just a matter of, okay, if they've already got this relationship, maybe they could come up with something with the spent yeast as well. Because yeah,
Okay.
that would be win-win for the farmer and for the brewery. Absolutely.
Okay, thanks.
Mhm.
Great, thanks, Jill. Looks like we have a hand raised. Greg, would you unmute yourself?
Thank you. I just want to say I really enjoyed the presentation, so thank you.
Thank you.
You already spoke a little bit about this with the other question about trials, feeding trials, which was one of my questions. but the other one was about, I think you referred to the brewer's yeast or the leftover yeast as a supplement. How does it compare with, and how would it be combined, do you know, with other feed that farmers usually use with livestock?
Right. So the nutritionally, the main thing in yeast is primarily protein. And so I can't speak at all to animal nutrition, so to speak, but so if you think about it, most of what the cattle are fed is hay. And if that's the case, how much protein is good for them, how much is safe for them, that sort of thing. So it would be, you'd have to ask, kind of an animal nutritionist, okay, if yeast contains this much protein, how much can we safely give them, so that they'll eat it and that they will turn it into either milk or meat. So it's, I guess, yeah, does that answer your question?
Yeah. I'm just curious if it would replace completely whatever their feed they were given, or like you said, would it be a supplement?
No, it would definitely be a supplement. Because they would still need to eat mostly plants, but it would be a nice way of giving them both some extra protein, and potentially these hop acids could help improve the efficiency by which they got the sugars and the proteins out of the hay or the grass or whatever else they were eating.
Great, thank you.
Yeah.
Okay, we have some more questions from the chat section. Ashley asks, has there been any attention to spent grain from distilleries in which craft yeast would be present but without hops?
Sorry, could you read that once more?
Absolutely.
Mm-hmm.
Has there been any attentions to spent grain from distilleries in which craft yeast would be present but without the hops?
So I'm not sure that well, I'll start answering and we can do a follow-up. So if, oops, I'm going to go back to my early slides here. So spent grain There we go. So spent grain is removed from the process before hops are ever added. So all of the spent grain from any brewery, be it a craft brewery or a Budweiser or whatever, it's never seen hops. So that I don't think is an issue. I think all spent grain is more or less created equal. I mean, certainly barley is different than wheat, but I think all spent barley, all spent wheat is the same, regardless of whether it comes from a craft brewery or from a multinational. And if that doesn't answer your question, yeah, just ask a follow-up, please.
Yeah, you can, Ashley, if you want to, you can feel free to unmute yourself. Give you a second there. If not, we have another question queued up in the chat. Okay. This is from Elsie Martin. She said there was a slide with brewers named blinded. What was the competitive information that would have been disclosed?
A slide with brewer's name. So I guess this, I know I mentioned it on this slide. So when the brewery makes the beer, they know, okay, this is how many barrels are, this is the volume of the fermentation tank. So we need to add, this many pounds of hops to it. And so that is generally very proprietary information. So I think, so that this is the main one where they didn't want us to show any of the exact hopping rate. So we can say, okay, oh, that someone makes a beer that has, however many pounds of hops per barrel.
Great. Do you have any follow-up questions on that? You can feel free to unmute yourself. Okay, if not, we'll be moving forward with the next question. We have a hand raised. Deborah?
Hi, thanks. Great presentation. My question is more about applications. So if you could dream big, like what gets you up in the morning besides your beautiful family who I'm seeing on this screen? What gets you up? What do you think about in terms of if you had your wish list with how this would be applied?
I mean, so ultimately, it's sort of like if we, again, thinking about this slide here, right now, there are tons of breweries in Asheville and around the nation, around the world, that are making all of this spent yeast. And most of them are either are composting it either in a pile out back or in the sewer system. And it's kind of like, we know that yeast is a complete protein. It's a great protein supplement. Humans won't eat spent yeast because it's it's way too bitter. It smells nice, but it's way too bitter. But ruminants have no problem with bitter foods. So it's sort of like, there's this, I see there's this material that's right now treated as waste. It could be used, even if it had no antimicrobial effects, it's got protein in it. That's a useful feed supplement. So is there a way that we could process it so that some animal or some to repurpose it other than just, you know, dumping it down the sewer? And then it's sort of like, wow, these hops have all these sorts of interesting antimicrobial effects. If we could, you know, I don't know, I think this gets back to someone else's question, you know, about how much of this stuff is actually produced. But, you know, if we think about how many cows are in the US versus how much spent yeast is produced, hoppy spent yeast is produced every year. If we could get this spent yeast to the farmers with the cattle, they now have a new food source that the breweries don't have to pay to dispose of. Maybe they even get some money, a little bit of money from the farmer to get it to them. And the farmer has cattle that that are more feed efficient. They don't have to feed them as much to bring them to weight. And so how much methane will then that keep out of the environment? So that's kind of the big picture of why I'm excited about continuing to work on this project, because it really is taking trash and trying to mitigate global warming with it.
Thanks.
Great. Thank you all for your questions. I think we're going to wrap up here as we're approaching 7 o'clock. Get some final words from Elisa.
Sure. Langdon, thank you so much for sharing your research with the members of the Asheville Science Tavern. And in my introduction, I was so excited to hear you speak that I forgot to introduce the fellow members of the Asheville Science Leadership Team who are here. One of them you already know, Harris Wagner, who has been moderating the chat. But we also have Dr. Jill Yeager, who is also here, as well as Dr. Stephen Martin, who's an astronomer. And did I forget? If I've forgotten anyone else, please let me know. But we're always looking for members of the community who are willing to share their scientific expertise with others in the community, and also members of the meetup. If you have ideas or subjects that you would like to hear addressed in these second Sunday for science events, please let us know. And with that, I just want to give another round of applause to Langdon, and thank you for being part of our group tonight. And I'm just going to wish everybody a good evening and sign off. Did I miss anything, Harris?
Nope, that covers all our bases.
Happy birthday to you.
Thank you.
Start next week.
We will be having a YouTube channel up soon, so if you want to revisit any of this talk, it will be up there shortly, as well as our last talk that was online. So just FYI.
All right. Thank you, everyone. Have a good evening.
Thank you.
Thank
Bye,
you.
everyone.
Thanks, Langdon.
Bye.